Not a fan of the wet hair Kyle look. Not a fan. It'll dry out eventually. Anyways, welcome everybody to the Roach Wiggle February. February had a long discussion about how to pronounce this month a couple of days ago. February 2nd, the glorious year of 2023. No stream issues. Just started things off straight from using Microsoft Edge. I think I need to get rid of Firefox or figure out how to get Chrome without the whoever owns Chrome from stealing my personal information. Blah blah. It was really wonky today. Maybe I should not have combed it back before I started the stream. So let's see who there is to say hello to today. Tennyson, Chain, Exotic Wilderness, Altered Pants, Tennyson again, and Jojo Macro. Hello everybody, and Braden. Good to see everybody. I'm sure the crew will grow a bit more as the stream goes on. I have been keeping the inbox pretty clear lately. I have one email that I want to get responded to. I'm gonna pull a Kai and do a flex, a low key flex though. Maybe I'll answer this later because it's that's gonna be they're requesting some husbandry information stuff. Harvard, just Harvard. Wait a minute, Harvard, where Roth's cockroach collection was. Where those hissers that I'm trying to get a start of are from. You know what? I'm glad I did this because this is making me realize I need to tell this person I need some things from this person, possibly some information. So I'm glad. Okay, I'm glad that I said things when I did. What? My hair so messy. This is distracting. I'm also wearing a different hat today. For some specific reasons. Last year, I think I had talked up on the stream about doing a fish day, and wherein myself and some other familiar names went around Southeast Michigan to visit some of our very abundant and very quality fish stores. Surprisingly, of all places, the Detroit metro area has a good number of quality fish stores, and to this day still has them, despite a couple of the ones that I grew up with closing. So we're making the day of that on Saturday, and it sort of spiraled out of control. It was gonna be just myself. It was gonna be me, Brandon, who hasn't been on the stream before. I mentioned before. He's worked here a couple of times on and off. I haven't really necessarily had him on here for. I may have him as a guest for doing like invertebrates in education, since that's his big thing. Brandon, Will, and Kai. That's who we rolled out with last year. Agnara madagascariensis. I'm going to have to update my label. Sorry, my eyes just drifted over here to Tennyson, all this information. I have them labeled in my colony is labeled as Agnara species. Thai, it's nice to see that there's a little more formal information there, and even more interesting that the Latin implies that they're native to Madagascar. I don't know if that's a deceiver like with Paraplenia americana. But anyways, so it was supposed to be just the four of us, and then I have a friend from high school. I don't know if I mentioned him here before, Mark. He flirts with the idea of raising beetles and is romanticized the idea of beetle fighting, and he is notoriously kind of difficult to get to do things. Hello, some eggs. But anyways, we're supposed to go over to Kai's place and look at some beetles before we go out to the aquarium places. And so my friend Mark has decided that, he's going to actually show up this time. So he's going to be there. Then we thought, we have a friend Tom, who I used to work with when I did the native plant stuff. And so invited him. He ended up being free. My neighbor down the street, Joe, who I will be doing a stream with at some point where we, when the weather's better, where we go out and look at his man-made pond, his swimming pond, which he's put a lot of time and effort into. And we will do a stream there eventually, look at some of the insect life and whatnot. Them, who else? Alan, Kai somehow convinced Alan to fly to Michigan, which Alan hates Michigan because it's cold. He was cold in Michigan in August. And so Kai has convinced him somehow to fly to Michigan for the weekend during some of our coldest weather so that he can go to the fish stores with us this weekend. And then Brandon wanted to be an agent of chaos and mix a bunch of people of different hobbies, interests, ideologies together, and asked me to invite a girl that I went on a date with. We've agreed to be friends mutually. To invite her because she is kind of vaguely interested in this sort of, well, not really vaguely. She's very interested in wetlands, and to show her some beetle. To go and see beetles with us at Kai's house, and then to go to fish stores because that's kind of her thing also. So we've sort of like not everybody's going to stay with us the whole time, or not everybody on that list is going to be there. She's busy. I don't know how long Mark will stick around. But we've basically sort of gotten this like massive group of people who are just going to drive around Southeast Michigan on Saturday and flood into these fish stores to just look at what they've got. And so yeah, it's gonna be. I'm really looking forward to Saturday. It's gonna be nice to get out and do something. Fish stores are always nice because they're always hot, humid, lots of cool stuff to look at. Sometimes they get, they've got weird microfauna living in their tanks. Like that's where some midges that I have came from, was from the local fish store. I've been in love with those things. So yeah, it's gonna be. I got this is my hat. This is my Christmas gift from Brandon. That I have to wear all day Saturday. So I'm really looking forward to this weekend. I've got a bunch of stuff I need to wrap up. Not really a bunch of stuff. I've got some scheduled husbandry stuff I want to get done tonight and tomorrow, and then I'm pretty much I'm freed for the weekend until next week, which is going to be excellent shipping weather. It's going to be in the 30s, 40s. I risked some boxes this week. Don't let Chris Snyder overhear this. I risked some boxes this week. Priority mail. It's been so cold and windy and stuff, and I guess I've just kind of, not that I've stopped caring, but I've just kind of been like, past couple of years, express boxes, some of them get there days later than they're supposed to, and they're fine. And some priority boxes I've sent in years past, the last couple of years in the winter, have been fine. So I tried a couple things priority mail this week that I would not normally have. And so far, nobody's reporting any DOAs from the bunch of orders that I sent out. I did delay some things to next week. I don't feel comfortable sending mantids through priority mail. It's a combination of how I pack them. I could and should pack them better. It just takes so much more time, and I'd have to pass that time cost on to people buying them, and I don't want to do that. So it's easier to just have people upgrade to Express. It's better for the mantids anyways. So, but some roaches, jumping spiders, isopods, etc. I did priority, and it seems to have worked out. So I'm very happy about that. It may end up being a game changer to be able to ship things less expensively during the winter to a lot of people. So let me do a quick catch up in the chat here. Exotic Wilderness asks, question about Uricos floridana. Probably wrong name. Are most of them red or stay a black color? Floridana is the one that is usually maroon, sort of like a dark purplish red. Lixa is the one that's solid black out of the two of those. Chain asked about beetle fighting. Are beetles that aggressive? It is an age-old tradition in Asia to fight various types of beetles. It is an age-old tradition in Asia to fight various types of animals. But with beetles, most of the time, with most types of beetles, it's not fatal or anything like that. Dorcas can really mess each other up, but a lot of beetles have evolved very specific ways to fight without becoming mortally wounded. So a lot of their morphology, the angles on their body, on their heads, on their elytra, on their legs, etc., are very heavily selected to sort of let the opposing beetle of the same species armaments slip or slide off or not be able to get a good grip. But yes, so I have an interest, not so much, I would like to fight some beetles. I'll come out in public and say that I would like to see if hissing cockroaches could be done. It's not, especially with roaches, it's not fatal or anything like that. And just having a mixed colony, they're inclined to rough each other up a bit anyways. But it's an interesting cultural thing, and I'm interested in it. And yeah, so Dorcas and things like that I've been getting into and keeping and whatnot. A lot of lucanids, all the beetles with basically armamentations are species that will be antagonistic with other males. Not always. Depends on the temperature, how long the adult male's been out of its pupal cell, a bunch of other things. Brady asks, over under on how many times Alan will complain about the cold. Probably every time we walk outside. So probably over 15 times, I would imagine. There are some humorous videos on the internet of stag beetles getting flung across the room in a fight, says some eggs. There are a couple of videos on YouTube I've seen of people coming across beetles fighting in the wild. Take it with a grain of salt. They could be staged, but they're kind of like one-off YouTube video channels and stuff like that. So I don't really have a reason to think like, oh yeah, this person like, there's those stage photos of like a beetle and a snail find each other and they become best friends. There's all those like curated shots of like the beetle riding on the snail and there's like a puddle and they like ride a leaf over. It's like, no, that's complete crap. But like these are grainy YouTube videos of somebody like walking up to like a cut over tree and there's like two beetles on the sap and they're locked together and stuff like that. But yeah, I think that one that I'm referring to was Dynastes Hercules, and yeah, you see the two males locked together and they're trying to flip each other off and they're just making these horrifying noises as their tarsi are like grabbing and pulling on the wood as they're both trying to throw each other off. Yeah, picture male hissers pushing each other off a platform or something, says Braden. Yeah, it would be, and very clearly also with beetles too, is when one of them has had enough with the situation, they'll just back off and run away. I've definitely seen that a lot with hissers when they are out pushed. I hear the boy at the back door, so I will be right back. So let's see. I caught what you said, Tennyson, about the Cubara species Borneo being Murina. That's not very surprising to me. They are very similar to each other. Yeah, I wouldn't be surprised if Panda Kings and Marina are very closely related too, based on morphology and life history. Chain commenting on how Blatella germanica males are very antagonistic to each other. Some of those things are density dependent too. At lower densities, things will be more antagonistic, and at higher densities, they won't really care. It is interesting. I wonder about some of those things in the wild, how that applies. Like Simondoa males, adult males regularly get their antennae bit off in my colonies, and the females are unaffected. So there's definitely something going on there. But the question is, like, how did that play into their natural history. Like, what was that an adaptation for, or exactly what was going on there? I don't know. Tennyson brings up about Levis orange not being Levis or being a subspecies labeled as CF Levis by Nathan. We had discussed that. I had brought up to Nathan. He had been keeping dairy cows with something else and had commented how they had crossed. And then I was talking to him about how, like milkbacks are not. That's not a thing. The milkbacks are a het for a Dalmatian phenotype in that group of Levis. And he was like, oh, and I was like, yes, I've kept like Levis orange and dairy cows. Lots of people have kept the two of them together and they never ever cross. So there's Nathan's doing some great work on stuff, figuring things out. If anybody's going to work through the messes of all this isopod stuff, it's definitely going to be Nathan, whoever he's associated with. I have put forth to Nathan, because it's looking like the Western Venezillo Arizonicus, the ones which people were calling for a while Californicus, even though I don't know where anybody got that from. One of those things that somebody just said and everybody kept saying, kind of like Labrus fusca. But Californicus is not a valid taxon or anything like that. They're just California or Western Arizona Venezillo. But I had mentioned about how there's a group of us boys who have been fisting the burrows and going to those places to collect those. And I had humbly requested that, should he formally describe the Venezillos from Western Arizona, I had requested that he transliterate the Latin to something along the lines of Sandy Boys or Sand Boys, in sort of honor of all of us who have gone out there, all of us boys with an I who have gone out there and fisted burrows and gone through the sand looking for those guys. I think it is a suitable name, at least in reference to the sand. The boy part is negotiable, but we're going to see. He's actually taking a trip out there in a couple of weeks, I think. I would assume he's going to collect holotype specimens, but I don't know if he'll actually find them at this time of year. So, so I'm hoping that I'm really hoping that he takes that suggestion to heart. He and I have been discussing them a bit, so I'm hoping that my sort of input on the situation and Alan's and the other boys with an I involved will sway him to give them Sandy Boys as a Latin name. I think it would be extremely fitting for them. Some Egg says, I found that female red runners will not necessarily be aggressive, but they will stand in their food to the point where other roaches can't get to it. I've seen them pick up food and run away with it quite a few times. A couple of other bladdids I've seen do that too, when they're really hungry for stuff. Tennyson says there's been a lot of progress in name updates and whatnot over the last year. Nathan had told me that he's working with the U.S. government and do not take me word for word on this. This is a summarization of what I think he has conveyed to me. But basically, the government's like, we don't know what any of these are, and we don't know what ones are potentially hazardous or problematic to the environment, and these aren't even labeled. We need somebody with expertise to give some input on this to sort of create legislation and guide legislation on stuff. And so I believe that's what Nathan's been working with, because as we all know, isopods have been coming in left and right for the last 6 years from all random parts of the world with reckless abandon and disregard. And so it's interesting now. There's the legislation is sort of grabbing for straws at what to do and how to properly manage things in a responsible and environmentally and agriculturally sustainable and informed manner. But that's none of my business. So anyways, other updates, other things that have been going on Fish Day is the big one. I'll have a lot to say next week on that. Anything going on here? I've moved my pseudoglomerus back into a enclosure that more closely resembles the one that I had a bunch of success with them in. I moved them into something larger. I wasn't giving them the more personalized care they seemed to really thrive with over the summer, and I found myself sort of desperately grabbing at getting the culture going back to a good point. Somebody asks, just curious, are there any plans to show bug tonight? Yes, I will. I will show some bug tonight. Imanis says, handsome over already, winky face, tongue out. Yeah, I'm trying to pull them out of the spiral. I'm hoping they've been sputtering out a couple of babies here and there. It does not inspire confidence. It's not like last year where I got those videos of the female just loaded with babies. So I'm going back to what worked and hoping that it's not too late. And so I'm going back to really keeping up on giving them fresh apple, which I think played a big role in things. They were eating fish flakes, dog food, and apple. I think having fresh apple available was a big part of it. Decreased the enclosure size, not so much that it's as small as it was last time, but enough that it's not the big 10-gallon I sort of threw them into recently. So I'm going back to sort of misting them regularly. I will probably try and mist them twice a day currently to sort of simulate. Last year I was very on top of it. Basically every time I walk by the cage I would give them a spray, and I'm wondering if that really made a difference, if it's something that bumping the humidity by trapping it in really can't replicate. So we're going to hope. Meanwhile, Pyrospherists have been doing really well. In fact, I'm going to take my culture off of this rack and set it aside because I want to bump them to a bigger enclosure. It was cohabiting them with these, what did TJ say this there? Margatia CF bisignata from Macao. And so I have a cohab of these two species, and the Pyrospherists are doing spectacular. I am so ready to have an actual colony of Pyrospherists going again. Pygmaeus, getting them going again has been a nightmare, and these punctatus have been really easy. I'm getting babies from ones that I raised. The babies are growing up, and so it's time to put them into a larger enclosure and hopefully have them available regularly. And then we can have these guys regularly available, which would be nice to have an actual roly-poly roach available very regularly for the first time in a long time. Maybe one day I'll get a big group of Pyrospherists again, of Pygmaeus. I don't know. We'll see. But anyways, for now, I'm very happy with how these have been doing. And I mean, if anyone's saying Margatia, these are also thriving in here. So another ectobioid. Put it on ectobioid roach. Put it on the pile of ectobioids. So put that there. I've got some megaphasma hatching already. I was not ready for this. This is months early. Fortunately, again, red cedar is evergreen, but now I'm going to be tasked with keeping them hydrated while also keeping them hydrated and well ventilated. I'm kind of grasping for ideas on how to do that, but I'm just going to jump into it and throw them something by the end of tonight. I don't know what it's going to be, but I just want it to be done. Tennessee asks, have your Masola hisser males been pretty solidly patterned? I sent a photo of some of my males in a chat TJ was in, and he pointed out mine were super cool compared to his, I guess. I've had a good mix of solid with some striping and some that are very heavily striped. And so it's been a pretty good mix despite them all being in one litter. And I could have just gotten the luck of the draw with genetics from the original pair that TJ sent me. I requested to keep some phenotypic diversity. I requested some extra individuals from TJ. He sent a couple, and I paired them with dissimilar individuals from the ones that I raised. And As soon as I confirm babies from those, then I will be moving some of the Masolas out of this culture. I will probably move all of them out of this culture into the main culture downstairs. And I just did this to keep some phenotypic diversity. I do not care about inbreeding. I just wanted to make sure that we had a good diversity. The colony had a good diversity of patterns going into the future, so it didn't become too homogeneous visually, in case there were more you wanted to pick for conservation, the billion other things that I say. Chain asks about what's up with Salgenia raggii. I wrote a blog post on those, which I think you're probably referencing. I have not yet bred any Salgenia. I'm cocky enough and confident enough to say that I will. It's a matter of time. They're doing very well in the setups, and having bred Panethia and Encaudelia without too much hassle, I think it is just a matter of time. But I just thought it was so cool how they were following each other. And again, there was a sort of very intelligent way to their movements, something that I have not really seen in other invertebrates before, where there seems to be a high degree of brain power to this one very specific thing, and that is probably following each other, staying coordinated in this way. And so it was some they turning into termites, says some eggs. The other experience I had with that was with a vinegaroon, and the way that vinegaroons feel, especially if you pick one up and it's uncertain about what's going on. It's a very human, it's very intentional how they feel around. And I was very enamored with that. And this Salgenia situation reminded me of that, seeing them crawling through the tunnel, and they would sort of all stop together and just sort of feel around and coordinate a little bit of movement, and then they would start moving again. It was very impressive to me. Jake McKenzie, I don't think I've seen you before, so welcome. Asking about the all-purpose roach and ice pod substrate. It's a very simple mixture, but I will say I go for high quality ingredients. It is coconut fiber, coconut croutons, sphagnum moss, some sort of hardwood mulch, and I know I'm missing something else in this, and well-rotted leaf litter mixed together. In about 50% of it's coconut fiber, and the rest is about a 1-to-1 split of all those other ingredients. But I try to get the highest quality stuff that I can, so the leaves are from the leaves that I soak the tannins out of for many months. And the coconut croutons are high quality hydroponics ones that don't have a lot of the fiber and other really cheap, not effective for bug substrate fillers, like the little strands, the strings of coconut stuff. A lot of bugs don't really like that. And then good coarse grain coconut fiber, which you don't always get. Sometimes you get the stuff that's like really powdery, and a lot of stuff also doesn't like that. Jordan S., welcome, because you're saying you finally caught a live stream. I want to keep some native roaches in the spring, and Parkoblatta virginica is the only common one near my home. Is it easy to keep and breed? Yes. Asterisk. Parkoblatta virginica are very easy. You can keep them on a mulchy substrate. You can give them bark. They will eat fish flakes and dog food and apples and all kinds of easy stuff to give them. The only thing is that depending on your location, there's a good chance that they need a winter diapause. So if you want to keep a colony going long, like through the winter, because all growth and reproduction will stop. If you have adults in the colony, when you get towards winter and they've bred, they will still keep laying eggs, and eggs will still keep hatching, but you will end up with a pileup of subadults at some point for a lot of localities, and you need to cool them down for the winter to get them to close into adults and continue the cycle. And this can be really easy depending on how you look at it. If you're a roach breeder like myself, and cold space can be at a premium, or it's kind of annoying to throw something into a cold place over the winter, then it's like, I don't want to do this. Like, I just want to leave a colony at room temperature and have it keep producing babies forever and not have to do anything. But some people, If you have a garage, a well insulated garage or something like that, or if you don't feel like, putting or if you feel like putting a roach bin somewhere cold, or a spare bedroom or something like that, or your basement, you could just throw the colony into that space for like 2 months, and then pull it out, and you'll get them to keep going. And some people, a bit of a break from having to do husbandry, I suppose. Braden asks, any Andromana litters yet? Curious what the gestation time will turn out to be. Nothing as of the last time I checked. And I've talked with TJ. I don't think he's gotten anything since the last time we talked. I think he would have messaged me and been like, I got baby Andromanas. From the original keeper, it sounds like there is a ridiculously long gestation, but this is getting to be like beyond the point of crazy. And I'm almost wondering if maybe I'm keeping them too warm. Supposedly they come from some cave system in that area, and so giving them heat like I've done with other hissers may not be what they want. I may run that by TJ. Maybe I'll turn the light off, or I'll just throw them downstairs in the bug room at bug room temperature in the 70s, instead of giving them the supplemental heat. But I don't know. I'm annoyed. I'm wondering, maybe it could be humidity. Maybe they're just gonna drop some babies when March swings around and the humidity goes up again, despite all my watering and stuff. So, I don't know. We're gonna see. I want a big colony of them too. So, but I have to sit on my hands. The supposedly pure stock that TJ also sent me a couple of last winter at about this time, that has been producing some babies. And I think it's pure. I'm gonna wait until this next generation matures and look at how consistent they are. But I'm feeling a little bit more confident that they may be pure stock, and I guess we can always have them sent off to Europe, or that one person who's supposed to do the phylogenetic work in the US could look at them and see if they are pure stock. But if so, that was easy, because they're doing very well for me right now. Chain asks about Meliroidia in culture. Oh man, I have to use Microsoft Edge to figure out what these are. Oh, those guys. Oh yeah, with that hack paper about, they had a white queen with them or something like that. Oh man, how has this not been thrown out yet? Jeez. September 2020, and they still hasn't been thrown out the window yet. Yeah, they look awesome. I don't know when we'll get them into culture, but they are a beautiful species. But man, I can't believe that whoever was like looking at this was misinterpreting the behavior so hard. Like, I don't know, man. I mess up a lot, but like, before you go and publish a paper about something as big as eusociality, you should really like consult somebody with even like a basal understanding of cockroach like life history and natural history. Because seeing a 10-year-old adult and thinking that a bunch of males swarming it is some sort of eusocial behavior around a queen is just so, it's like the equivalent of the person who goes on like any forum is just like, I found a white cockroach, is it an albino? It's like, oh, you sweet summer child. Like, yeah, sure, teach the person. There's nothing wrong about asking questions, but don't publish this. And then it's still up with all these pictures, plain as day to see. Oh man. Some egg says he's found some of his P. virginica nymphs dead with a fungus on them. Not sure if the fungus is necessarily killing them or just colonizing the dead. What color is the fungus? Some eggs. There's was trichoderma is bright green, and that does kill the roaches themselves. It will sometimes also colonize roaches that have died. Tennyson asks, how long has it been since you guys got adults? I think I got the adult Princessia back in June, July. So this is going on a very obnoxiously long amount of time. And the female had definitely created an ooth at some point. So it's It's you know, like it's not like, oh, they waited this, she matured, her ovaries still needed to mature and then begin producing eggs. She hadn't made an ooth yet, so she's going to make one. This is just like in the same amount of time, we've already gotten like another generation of Masolas. It's just on such a different other level. So I'm wondering if there's a trigger missing or something like that, and we'll just have to keep our fingers crossed and just wait and see what happens. That's really all we can do. I'm providing them with excellent care as with respect to other hissers, informed excellent care as would cause most other hissers to enumerate into huge colonies in a small amount of time. So Jake McKenzie says, this spring and summer in New Mexico, there were dozens of Aranavega erratica outside my house breeding prolifically. Did you find females too, or just adult males? It's all that shows up on Google about them. Chain commenting on the Meloroidia thing. Yeah, I mean, they could have asked just about anybody. You could have asked anybody, any roach keeper about this anywhere in the world. But like, it's weird. It's like somebody viewing entomology through like a vertebrate lens or something, because it's like if I'm sure most of the people even in this chat, if you saw a 10-year-old cockroach, you'd be like, oh, it's just an adult, just matured, or oh, it's just with wings, oh, it just matured, it's just a recently matured individual. And the funny thing is, also in those pictures, the individuals around them have swollen wing buds too. It's like, did this person not know anything about like cockroach like life history at all? Like at all? And then there's all we already know about roaches that group up to amplify their aposomatic signals, which I would say almost certainly what's going on here. And that's why there's the redhead Oxyhaloa do that. The adults have their defensive secretions and whatnot. In fact, it's almost a 1-to-1 in terms of like, Oxyhaloa, I have a bright red head, a dull purple body. They're in a mimicry complex, and they group up as adults and whatnot to amplify their aposomatic signal, at least of the adults. The idea being that like the adults having those colors would be protecting any of the nymphs around them that have not developed those secretions yet. Or then in Deropeltis paulinoi, and I guess in the Masai Mara and Jinka species, but most excellently shown in Deropeltis paulinoi, the nymphs are gregarious and they can be day active, and they will group up on the tips of wood and branches and bark and leaves to amplify their aposomatic signal, with the idea being that a predator may not get the idea if they see just one cockroach running around. It's sort of orangish and blackish. They may kill it. The individual may not have enough of the secretion to leave a lasting mark on the predator, and so they would just continue doing the behavior. Even if they didn't end up killing it, it wouldn't leave a big lasting impression. But if they go for a whole mass of multiple nymph individuals, then they can increase their individual survival rate as well as the group survival rate by releasing their chemicals all at once and really making an impactful stink. And then the predator will not disturb they will not disturb that species anymore, because they'll learn that they see this blob of orange stuff, that they're going to get this massive chemical defense. They're not going to mess with it, and thus it amplifies the signal and the effectiveness by being in a group like that. And that's what I think what I truly believe is going on with these I can't remember the genus. Here I'm preaching about how much I supposedly know about them. I can't remember what the genus was. Wherever it was a Melis something, Meloroidia. Yeah, I mean, the brightly colored, they group up tightly. It's almost like I would bet if you used an artificial intelligence to analyze the exact grouping pattern in these Meloroidia, I bet that it would be found to be extremely similar to, if not identical to the patterns that Deropeltis form when they group up as nymphs. They were both males and females, thought they were different. Were the females out on the surface, Jake? It's very unusual for them to be out on the surface at all. But also, if it was after a rain or something like that, it was humid out, and they felt safe, I guess, and there was a high population density, you could have a lot of females that would be coming to the surface calling the males. Very, very rarely have I ever seen Arena Vega out and about in the wild. I think I saw an adult female crawling across sand like once. I think I scooped a male out of the very entrance to a burrow once. Like it had clearly matured and had come to the burrow entrance to then take off that evening. I'm absolutely certain that's what was going on. So, but yeah, okay. I went on a little rant today. I was going to look at something. I talked about the Perispherus. Trying to think about other stuff going on. Stylopyga. TJ had gotten me some stock that supposedly originates from China. Is not as much of a pain as the U.S. stock, which is probably extinct, if not survived by the single female that I got out of some individuals that Junior had sent me maybe last year. But this China stock stuff has been doing really well. We'll see how they do long term. I think the key with Neostylopyga is that they want high humidity just constantly, and it's not a humidity that you can replicate by spraying frequently. It's you have to just keep them very humid with low ventilation if it's dry, or they just need to be humid. And another species that I think is like this, and why I feel kind of confident moving forward with my husbandry of them in this manner, is Paraplaneta brunea, ex Paraplaneta brunea. And so, because I received a group of those from Alan and set them up in my traditional roach enclosure, which is a hexagon with a couple of one inch diameter vent holes. So I've been keeping them warm and extremely humid. There's actually a touch of condensation in part of the enclosure at all times. And the Paraplaneta brunea, for the first time in, I don't know, maybe 8 years or something like that, are thriving. I have a bunch of nymphs. The adults have not even any antennae damage after being alive for like half a year or something like that. We have some in Paraplaneta missionary, and thus begins from Tennyson the antagonization to showbug. Jake McKenzie, I was watering my tree at night with a deep water irrigator, and they would crawl out of the hole I dug. That's fascinating. If you find more females next year, you should definitely contact me, and I would like to get a group from your locality of them. But anyway, so yeah, Paraplaneta brunea, and now I'm taking a similar approach to Neostylopyga to try and do some more of the same, and hopefully get a nice big old colony of them going, because they are beautiful roach. And yeah, that's that with those. It's been a while since people have really kept Neostylopyga in any significant colonies or numbers. The one person with a holdout of the United States strain, the old stock that came from Bugs in Cyberspace, was Island Reptiles in New York. And that's why I had recently, and by recently I mean like 2018 or something, restarted my colony of U.S. stock from. And then I had messaged him, I think, last year, and he had either lost or gotten rid of the colony. And so then it was like, all right, I guess they'll die then. Let's see, anything else going on in here? Nothing extreme to write home about. A lot of the Grylus are doing well. Any Grylus enthusiasts out there? I think there was somebody I was talking to over email who was very into Grylus. But I'm thinking that by this summer, I'll probably hit about 15 species. And as you all know, pretty much none of them are up on the site. I'm really hoping to do a site update in April, but I got to get through. I got a busy February. I'm gonna have a busy March. Going to be doing the Alabama trip for a week. This is going to be a busy March, and then that's also when my yard work stuff is going to pick up again. So I have to go outside and cut down the stems. You got to do poultry, pair up rabbits, move the guinea pigs back outside, do poultry stuff. I bought a huge incubator to hatch a bunch of chicks and stuff this year. Jojo says, my Grylus assimilis from you are doing insane. Love them so much. That's great. I'm so glad that ended up working out, Jojo, because that was, again, like, I was like, I don't know if these are going to survive this hell trip that they're going on. And they ended up doing it. And I would imagine you probably have babies by now, because I sent like mid-size or larger nymphs. Those should have matured, and the incubation period is pretty short for all. The incubation for all Grylus that I've kept has been very short. And except for, of course, the ones in I think it's the Rubripes or something group, which includes the fall field cricket, all of which are in like a taxonomic clade with each other based on the species that have eggs that need to be overwintered, as opposed to overwintering as eggs or nymphs. And that's a species I need to get this spring too, is the Springfield cricket. I really need to do that, because by the time that I'm like, I should go look for Springfield crickets, it gets about the point where the fall field crickets start having overlap in size with the Springfield crickets that have been born this year. So I need to go out in May next year and really put a good effort into collecting some Grylus velutus around here. They're very common in my neighborhood, but they're just kind of a pain to find for some reason. Over the years, it's probably all the predators that have moved in, like the cricket hunters and spiders and shrews and stuff in my front yard has definitely kind of kept a thumb on the Grylus velutus population. Pennsylvanicus is still very common, and I find them during the winter, but velutus for some reason is not the most common, as it is if I go down the street to my neighbor Karen or Joe's house, who both have their big native plant landscaped areas where velutus is much more common. And I want to just go out in May, collect an adult female, and then get a colony going from that, so I don't have to second guess myself. Paraplenia missionary asks, eggs crossing when? Some eggs crossing when, perhaps more appropriately stated. Tennyson, yeah, if you catch some Grylus this spring or summer, definitely contact me. If they have locality information, I would love to get groups, either groups of nymphs or groups of adults containing females, obviously. And I'd like to get them going. So, yeah, I've really grown to really like Grylus. Basically, once you get the colony going, you get that momentum. They're very hardy, but they can be difficult to start, because if you collect wild stock, you can bring in fungi, you can bring in velvet mites, you can bring all kinds of other stuff that will just stop you from getting them established, just dead in your tracks right away. But if you get them going, and you get up to a colony of around 100 individuals, it's very difficult, as long as you're providing food regularly, it's very difficult to lose the colony. They're very reliable that way, but you have to keep some degree of that momentum going to be successful with them. So, yeah, I would like to eventually have both the pictures of the adults, which allow them to look very similar but different sizes. I would like to have pictures of adults, and then also sound clips of the calls, so that people, if they would like to sort of soundscape their house, they can choose between different Grylus species and create sort of an ear feel for themselves. Tennyson, it could be, I wouldn't even ask you to identify species. It's difficult enough as it is. And there's plenty of taxonomic work that's being done on them, and splitting up all these species and finding all the clades and stuff that they're in. Again, with the spring and fall field crickets, it used to be under the assumption that they were very closely related and had diverged relatively recently based on their activity and their overwintering stage. And now it's been found that they're actually distantly related. They're from two completely different lineages of Gryllus. And I think that's really cool. I'll show some bug actually while I'm talking about this. It's not too exciting, but it's still showing bug. So I'm talking about Gryllus species. I have CF pennsylvanicus, but these are pennsylvanicus for sure because they survived being overwintered. So this is my hatching cup. I grabbed some females in the fall, really late. I think it was like late September or something like that. So it's far beyond the time that Velitas is active here in any form. You would not find adult Velitas in Michigan in late September. It's way too late in the year. So yeah, so I'm raising some Pennsylvanicus just for fun, and they'll probably be adults. If I give them heat, they'll be adults a lot faster. They could probably be adults by the end of the month if I gave them heat, but I'm just sort of keeping them at room temperature so they grow at a more natural rate. But this is just the egg cup that I use. I think you can probably see some of the eggs still in there. You can see the Secabria tropical pinks I put in there just to sort of keep things a little bit tidy. I have food tucked into the pine cone to keep it from molding in addition to the springtails. Crickets do not like moldy food at all. Yeah, I mean, you can see this apple's getting kind of gross. Has a couple oribatids on it, but they'll still eat it a bit. Once they get bigger, they can be quite voracious. So yeah, this is just it's just like, okay, I'll just have some fall field crickets. I'll just get a little colony of them going. It will be annoying because what this means is that when I have adults, when I want to get the next generation, I either need to set them up such that they can only lay in an egg cup, or I need to put them in an enclosure so that I can put the whole thing into the fridge during the summertime, because otherwise the eggs will not hatch unless they get about, I think in the past I did as little as a month of cooling, but I think that ideally you want to do 2 months of cooling to get Pennsylvanicus to hatch. So yeah, so this is just a little fun Gryllus thing here. Tennyson, these are Gryllus pennsylvanicus, just fall field crickets. They're very common in the eastern United States, especially in the north. Periplaneta wants me to show Periplaneta. Unfortunately for them, he's limited to 2 species in the genus Periplaneta currently, neither of which I have in this room. Gryllus pennsylvanicus, when I really went down there. Don't you have pennsylvanicus in your area, Jojo? They range all the way up into Canada, but maybe not in your part of Canada, though. But they should. Jake McKenzie asked about fermenting dog food for roaches. I've never fermented it. I always just give it to them straight as it is. Tennyson asks, is there such a thing as a silent Gryllus? I think that there are a couple of species that might have gone silent in response to parasitoid pressure. I don't know enough. I know that there are taxa of orthopterans that have gone silent because taconid flies and other stuff usually use calls of their host to locate them. So I'm pretty sure there's a silent Gryllus, but I don't know what it is, and it might be quite rare. It might be one of the ones from the Florida Keys or something like that. One of the endemic ones in the Florida Keys, which I would really like. Oh, actually, never mind, Mr. Periplaneta. I forgot that I brought these up here. These are for a breeding project. And I'm waiting for, these are all virgin females that I picked as subadults. I'm waiting for them to mature so I can see which ones are most similar to the male that I'm breeding them with. But this is a special strain that Alan found. We're really hoping that the pattern that the male has is heritable. They have a completely unmarked pronotum, at least the male does, and reduced markings on some of the individuals. But this will be the real test, is whether or not it proves out when I take the male that sort of founded the stock and cross him to his daughters. These are all his daughters. We'll see how things go. It may end up being not heritable. It may just be, just maybe a big old flub all around, but I'm going to really hope that it's not. And the name of the strain, if it is heritable, is Baldy Bear, for a bunch of reasons that I don't want to talk about. So there we go. We showed some bug. I have one more I'd like to show before I wrap things up, which won't be too long for now. A little bit more though. Oh, Shane, you got me excited. I thought you were about to say that you found a bunch of young colorless Pluraloma flavipes in one of your enclosures, but that's exciting still too. Jake McKenzie asks, he asked about fermenting the dog food because some people have done it with chickens to cut down on feed costs. I have a friend who does that with her chickens. I don't know if she still does it though. I don't know how to do it, but I've been pretty content with the amount of food that my chickens have been getting by on, and all my poultry through this winter. I've been pretty happy with it. Actually, it's been the pigeons who have been really putting away the food, like so much so that I'm like, is this really a smart move for me? Like to have these pigeons eating so much? But I think they can get by on just corn, so there's that going for them, I think. But still, Shane, what type of log did you find them under? Was it white rot? Was it brown rot? Was it really moist? Was it really dry? Was it There's a lot of things. We're still finding our way with all these flat millipedes. Someday somebody will find something that you can just throw at them and they'll thrive on, or we'll figure out what their humidity needs and stuff are exactly, what these things need one day. Cool air for Pluraloma. Now, I can't remember if it was Pluraloma or another species that I collected locally and saw at a local state park. This is back in 2017, I think. Kim took me there. It had rained very heavily, I think that night or the preceding day. And I want to say that it was in May or June. And we went there and we were, just walking through. It's like some of it's like surprisingly older growth forests in parts, but it used to be like in sanitarium or something like that in that area. I don't know exactly how that worked because there are a lot of natural features there, but maybe they just built the facilities into the woods or whatever. But I was able to see. We were really curious about what they were eating. The flat millipedes again. I think it might have been Pluraloma or it might have been something else. But on that day, they came out and they were everywhere. They were crawling up the tree trunks. They were on the paths. They were in the leaf litter. They were all over the place. And I was able to sort of get down and look at them and see what they were chewing on. And they were very polyphagous. They were eating the wood. They were eating the moss. They were eating the lichens. They were eating the leaves. They were just sort of eating a little bit of everything. And it wasn't cold that day either. It was in the low 80s or high 70s. So I don't know exactly, what combination of what we need to have to get these things to breed. It's possible maybe that they could be kind of like a parkablata where they need to be cooled when they're subadults or immature, and then they need to molt into their adult or mature phase, and then they reproduce and they die. I'm not sure. Yours have been so straightforward. It just makes me think like, these other things, it's just got to be something. You just have to find You just got to find their thing. The pseudopolydesmus were easy. The Euryurus were easy. It's just like, what is their thing? Pachydermis. Alan had excellent results getting pachydermis to breed, and I don't think he ever figured out why the ones that he raised about halfway started dying. I think he said he just didn't take good care of them. So if you stop giving them more fermented wood or whatever, and they run out of their other stuff, they'll just starve very quickly. If you think about it, it makes sense because in the wild, there's no limit to how much leaves and wood they can find. Like if they run out of if it gets too dry, they'll just estivate. They'll just curl up in a little poop chamber underneath a log and wait for it to get moist. And if they've run out of food at the log or area that they've been feeding at, they just move somewhere else. There's no reason for them not to. Nothing's going to eat them except for maybe a handful of carabids that go after them. But they'll just go somewhere else. It's not like there's a limited supply of all this stuff in the forest. And if there is, then the population probably just disappears. At U of M in the natural area, I don't know exactly what they like to call it nowadays, but I always call it the natural area. Let me see. U of M natural area, just so I have something to tell people. Okay, they're calling it the ESA, the Environmental Study Area, University of Michigan Dearborn. In the Environmental Study Area, every couple of years, the land resources, the land manager, etc., Rick Simic, who is one of my huge inspirations growing up and a lot of my interests and whatnot, and I'll wax poetic some other time. Anyways, every couple of years, he'll find or there'll be a report of a flat millipede. I think they were Aphiloria or Sigmoria or whatever, one of the larger ones. And you would think in this pristine natural habitat that does have a lot of old growth forest because it used to be part of the Henry Ford estate, has a lot of big old trees. There are trees there that are hundreds of years old. With a diversity of habitats and whatnot, you would think that, there's all this organic matter. There should be these millipedes everywhere. But myself, having grown up there and having gone around the estate, all seasons, all weather conditions, night, day, wet, dry, after the 100-year flood, all this other stuff, I have actually never found one. And that was even in my youth. I never came across them with my dad when we go out there and flip logs and stuff like that. And so it's possible that some of these things, once they get fragmented, or once non-native isopods are introduced, non-native millipedes, non-native slugs, something that sort of competes for them for this niche of curling up in a poo chamber underneath a log and then running off to go graze a little bit on this type of wood and this type of leaf and this type of thing, that those populations could see gigantic, catastrophic declines at times. So it could be. They could just be a generalist that just is very general in a lot of other things and gets outcompeted or physically displaced by things that have been introduced or things that grow in numbers when the area gets disturbed or something. I don't know. But I don't think there's anything terribly difficult about getting them going. We got to just figure out what the thing is. So Alan figured out pachydermis. And people have been raising Euryurus and Arcturus for a while. Now we've got pseudopolydesmus going pretty regularly. So there's got to be something else. There's got to be something. I don't know what. Something. Are there any silent cricket species other than camels and Taphiliska? Yeah, again, there's a lot of in the hobby. It's not that I can think of really. Chain says not yet, but maybe I found the key to breeding flat millipedes. Maybe brown rot oak, presumably. Yeah, brown rot could factor into things too. It's also interesting that a lot of these taxa in the eastern United States are restricted to temperate areas. Once you start moving south in Florida and getting out of the areas that have some sort of seasonality, you don't find them. I'm going to look up what some eggs has posted here. Large whorled pogonia. I have never heard of it. I'm not much of an orchid enthusiast. I really like lamiaceous things. I like mints. I like seeing all the different localities of Monarda fistulosa here and what mints pop up in different places. Tennyson says I've also bred some local polydesmus species before. One of those was is a poly. It's that one that's on the west coast. I can't remember, Hyalaria or something like that. It's black and yellow. It's the main one that everybody talks about. Somebody was breeding those at some point. Somebody I was talking to in Texas was breeding them, feeding them fir leaves, and that made sense. It's like, yeah, it's raining fir needles constantly wherever they live. They can always find them in just the right condition that they want them. Hello, Gavin. Good to see you. I think we're all doing pretty good here. But yeah, flat millipede stuff. That is a passive target of mine when I go down to Alabama this spring. Is I will be collecting localities of whatever flats I come across. And some of them I will be working with. I will follow my whimsies. And if I find whatever locality or species or genus and I look at it and I go, this is really pretty, I want to try and breed this, I will once again give it a decent effort and see what I can do. And where was I going with this? Is the others I will be putting up for auction at the spring auction. Obviously, it will be made very clear these are wild caught. They've been in captivity for however many weeks or whatever. But to get them out there to generate some costs back from the trip itself, etc. But yeah, I think again, this is a Kim Wisman perspective that I don't think this came across from Kim when he was on. What was it, 2 weeks ago when he was here? I don't know if we necessarily got into this sort of style of discussion, but we talk about things like this quite frequently. But basically, it's like you bring something into captivity, you throw it in a box, and you feed it what you think it eats or what it's supposed to eat, and then it eats it, and then you fail with it, or it does well, or it doesn't do well. But you have to think about stuff like outside, there's no box for a lot of things. For things, they can go wherever they want to get things whenever they need them. You have to really consider the natural history of things to keep them in captivity. And with the millipedes, it's like, yeah, there's infinite food for them. If the habitat's right, they will never run out of food. They can walk 20 feet away to find the perfect flavor and perfect decomposition state of leaf that they want. And it's something you have to really work for and understand if you're going to offer that to them in captivity and be successful. So yeah, stuff like that. And I think millipedes being mini bioreactors really puts an interesting spin on that too. How they amplify common bacteria from around them from stuff that they eat. They don't really have a specialist bacteria per se. They just sort of amplify detritivore bacteria from the area around them and in their gut, and that's how they digest things. I think they will increase the density of bacteria up to 3-fold from what they originally intook. Somebody asked about how my pygmy dormice are doing. They're doing exceptional. They tear through roaches like there is no tomorrow, and they are also eating a lot of the fruit jellies that I put in with them. I don't think they've actually drank any water since I've gotten them. They've been just eating roaches and getting all the moisture they need from those and from the fruit jellies. So no babies. I think I'm due to clean their enclosure, but I have to go out and buy some more cypress mulch. I'm not going to use pine shavings for them. There's not enough open top ventilation in their enclosure for me to justify using pine shavings. So I'm going to use cypress because that's what they're on right now, and they seem to be doing very well. But later this month, if I end up going to the Cleveland Expo, which I may, because I may have to pick up some stuff from Oren there and meet up with him again. And I'd also like to go. I really like that Cleveland Expo. It has a lot of stuff to look at. It's a very good expo for me the last time I went. But anyways, I should be getting some African pygmy mice, which I've been wanting for a long time. They were kind of common between 2008 and maybe 2013. There was a little bit of a push to sort of get them to be like it's the perfect pocket pet because they're so freaking tiny. And then, I don't even know if you could call it a craze. Any residual interest there was in that just sort of dried up, and nobody had them. And so now I've been in contact with somebody who, it seems like people have to drive all the way across the country to pick up to get some of these mammals that people are just barely keeping in culture in the U.S. And so I have this contact who did just that. I think she got them from Texas originally and is now in Ohio. And so I'm going to hopefully get a pair or two from her. I don't really care how much it costs. I would just like to work with them. I think like a lot of other small rodents, they will greatly appreciate having roaches in their diet or just having insects in their diet. A lot of them really, 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 it's kind of like with fish keeping, where everybody's feeding stuff flakes and pellets, and then you see all the successful breeding reports, and like, we offered them live bloodworms, we offered them live brine shrimp, we offered them live this. It's all this live arthropod food that they give things before they have these huge successes with them. And so that's what I'll be doing with the pygmy mice, with the hilarious and fortunate thing about it being that a single adult female hissing cockroach could probably feed a pair of African pygmy mice for a week, which is just just crazy to think about. So funny. I think it was Mark, again, my friend from high school that I mentioned earlier, who had this really funny sort of observation or perspective on things. I can't remember what I was feeding. Maybe it was, or maybe telling him about how praying mantids eat like hummingbirds or something like that. And to him, it just felt so unnatural for like a lower organism to be eating like a higher organism like that. And the hissing cockroach thing with African pygmy mice kind of seems like a spin-off of that, where it's like it's this huge bug and these tiny mammals. I don't think they'll actually kill it. I'll have to pre-kill all the roaches that I give them, but it's just a very interesting thing to think about, these mammals that are so tiny eating this gigantic insect, basically. So that's that. That's my sort of little talk about that, I suppose. So yes, the dormice are doing well. I moved all my other mammal projects back in the laundry room. It's just so much easier to take care of stuff with a sink right there. It takes half the time to do all the cleaning and all that other stuff. So it was just like, okay, I'll put it on wheels so I can clean up when they toss their bedding and stuff more easily. And I'm very happy. I'm very satisfied with the results that I'm getting right now. So yeah, maybe one day maybe I'll talk about my mammal projects. I don't know if I can fill an hour time slot with just that, other than the boy and the rat. Okay, maybe I will do that eventually. Oh, definitely, there will be a Victor live stream sometime in May at some point, maybe around his birthday. I'll do a checkup. We'll have him in here. He's been very good today. He hasn't been making any noise or anything like that. He's been a good boy. So, hour and 14 minutes. So I think we're going to wrap it up now. I got to get downstairs and finish up. I'm doing my husbandry on the 2 beetle racks, husbandry on my miscellaneous rack that has a lot of my amblypygids and miscellaneous arachnids and miscellaneous bugs like the earwigs and the assassin bugs. doing husbandry on the sort of inventory bugs, things like the mantids and miscellaneous darkling beetles, millipedes, and some other stuff like that. Big announcement to anybody here. I will have some Idolomantis in stock very soon. If anybody here wants Idolomantis, it seems pretty reliably every 2 years I have an opportunity to get some Idolomantis. That opportunity has arisen again. So if anybody here is looking to work with Idolomantis, I will be getting some in very soon. This is your opportunity. It may not come again for another 2 years or so, but here it is. There will also be a mantid inventory update very soon. Following that, wherein I will be listing the Idolomantis. If you would like to get in on the ground floor before that happens, please let me know. I will spell it out here. Idolomantis diabolica, diabolical flower mantid. If you're interested, now is your opportunity to get some of them. Another one that's kind of like this is the orchid mantids. I have at least had them available a couple times a year. Unfortunately, the person who was primarily producing them in the United States died like half a year ago or something like that, very tragically. And that's just how it is, I guess. Hope that doesn't happen to me or anybody else here, but thus is the fragility and preciousness of life. So, but if you would like Idolomantis, again, get in contact with me, and we can go from there before things are listed and whatnot. But regardless, thank you everybody for tuning in. Next week should be an excellent stream with a lot of stories from Fish Day. I am really looking forward to it. Maybe I'll wear the hat again next week since I'll be recapping. This on my shoulder. Other shoulder. Oh, it's a hole, of course. So until then, everybody, stay warm. Spring's on its way. It's going to be in the 40s here pretty soon. And may your cultures ever be bountiful. See you all next week.